I think we are going to read last day and then last few talks, so we can relax a little bit. There's any questions during my talk, please feel free to stop me. So I would rather make it as a discussion type talks. To make sure that the content of this talk is clear to you, so I made some introduction. I know that some of you are familiar with epigenetics, so you may fall asleep for a couple of minutes. My talk will go over 3 sections. One is introduction to epigenetics, and the second is I'm going to talk about a new technique we developed. And third, we are going to talk about how to use a new technique and to do the data mining. So epigenetic basically is getting hot in the past couple of years. By definition, is heritable changes in gene activity, but without the DNA sequence change. So the sequence remains the same, but the gene activity changes. So epigenetic code can be divided into 2 kinds. The first kind is DNA methylation. So DNA methylation in mammalian cells is frequently happen on the CpG dinucleotide, on the cytosine. So the methylation is methyl group is transferred by DNMT, DNA methylation transferase. So we'll add the cytosine, the methyl group to the cytosine. Methylated DNA often found to be in a close chromatin region, heterochromatin region. So basically, the methylation results in chromatin changes. And DNA methylation have a partner, is histone modification. So we know that the DNA molecule is wrapped on the histone molecules. So it's around 146 base wrapped on the histone. And histone modification changing the charges on the histone molecule. Therefore, for example, there's so many kinds of histone modifications, but in general saying that some acylation on the histone will result in the histone to open up the chromatin. Therefore, gene transcription can initiate. And histone methylation, for instance, it may close up the chromatin region, and therefore you form a compact structure. Therefore, the transcription will get stopped there. Methylation, why is so important is one key thing. Thanks. One key thing in the methylation is related to the tissue development and cell differentiation. Now we talk about DNAs, but in our bodies, almost all the cells share similar DNA content, but they have different phenotypes. For example, your liver and your lung definitely is 2 different things. They have same DNA. So how they become different is go through the differentiation, tissue differentiation, and such tissue differentiation definitely is not changing the DNA sequence, but changing on the epigenome. On the other hand, in the past couple of years, is IPS, is induced pluripotent stem cells, development on the stem cells. So what they can do now is that they're able to reprogram the cell, which full differentiate cell, for example, fibroblast and lymphocyte. They can convert into the stem cell, reverse back by adding some transcription factor. And now ultimately this cell reprogram back, and now you can redifferentiate into the tissue you like. So the idea behind it is for stem cell therapy. So you can replace the damaged tissues. For example, brain disease, neurodegeneration. So something like that, you can replace in the tissue. So this is very attractive. So become very hot in the past couple of years. But either cell differentiation or reprogramming is rely on the change and the epigenome level. Now, I'm going to introduce the technique we developed. Before we start talking about the technique, I would like to introduce the bisulfite sequencing. So whenever you get sequencing here, I only show you CGs because this is not the target of DNA methylation. So there's 2 red is methylated and 2 blue is unmethylated. When you go through a chemical treatment called bisulfite, you will convert the unmethylated C into T. But however, the methylated C will not change. So by this way, when you do the bisulfite conversion and sequencing compared to the reference genome, you can find out what C is methylate, what is not. And here, just make it simple. People often use such a symbol to represent the methylation and unmethylate cytosines. For example, methylate cytosine using filled circle, and unmethylate use open circle. And this sequence reads the methylation level is 50, so basically half methylate. But half methylate can mean a lot of different things. Think about it. This is one methylation pattern that's half methylate. But there are different methylation patterns. So most our current DNA methylation analysis is talking about DNA methylation level, but rarely pay attention to the methylation pattern. In this talk, we are more focusing on methylation pattern. And I'm going to convince you, I'm trying to convince you how important methylation pattern will be to understand the DNA methylation data. So now let's look at the scenario. You now understand that each line here is a sequence read. For example, here has 16 sequence reads. And for this genomic position here, you see all methylate. Oh, whenever we do experiments, we are not there yet for single cell epigenome. So most likely you start with 1 million of cells and you just sequence cover all of them. So you get 16 reads here. So most likely each sequence reads came from different DNA molecule. So it's showing you here homogeneous methylation pattern. It looks like your population on a cell at this genomic loci. They are tightly controlled and have similar methylation pattern. But however, on some genomic region, you may see pattern randomly like that. So this indicates that the methylation is stochastic variation going on there. But some of the genomic regions shown to be very well structured, called bipolar DNA methylation pattern. But why you see bipolar DNA methylation pattern? Now I give you 2 DNA molecules. Then these 2 DNA molecules can come from 3 different resources. One, they came from same DNA molecule, but 2 different strands. You remember that the DNA is double strand, so it's plus strand and minus strand. So most frequent people believe that the plus strand and minus strand methylation is identical. But this actually is not going to be the case. I'm going to show you later. So this is the one scenario, is that whenever I give you a methylate DNA and unmethylate DNA sequence reads, it could be came from one DNA molecule, and then we call it asymmetric methylation. And the other scenario is that we know that from humans, you have 2 copies of DNA chromosome. One set is from father, the other is from your mother, right? So the 2 alleles could show a methylation difference. So this is called allele-specific methylation. And the third case, of course, you came from 2 different cells. These 2 different cells have different methylation pattern. So they have cell subset-specific methylation. So if you see 2 DNA molecules showing a different methylation pattern, this is 3 resources or their combination. Now, the allele-specific methylation, it has been studied for many years, imprint. And this has been known that in the mouse have 55 regions, and for the human have 51. So it's very clear known where they are in the genome. This is unknown. But asymmetric and cell-specific methylation, this is the target, the primary driving force to understand the DNA methylation, because we have cell-specific methylation, we have asymmetric DNA methylation, and how to tell them. This is the technique we developed recently that can tell the difference between the strand and between the cell molecule. How we do that is quite simple. We just add a hairpin linker to the DNA molecule. DNA is double strand. You link 2 strands together. Now when you do the bisulfite conversion and then you sequence it, so now you sequence from both ends. So basically you flip over this molecule come here and this go there. Therefore you can sequence from both ends. By this way, from the high-throughput sequencing, you can get both end information at the same time. So I'm talking about both strands. You sequence from both ends, but turn into 2 DNA strands, top strand plus and minus strand. So by this way, you can determine whether or not their strand showing any difference. We sequence mouse embryonic stem cell, we found that 12% of CPG site, the top strand and minor strand, they are different. So we divide the methylation into two groups. One group is asymmetric DNA methylation. The other group is cell subset specific methylation. Okay, because based on the hairpin technique, we're able to do that. Now we also include two groups as control. One is complete unmethylated. The other group is complete methylated. Now these four groups, CPG side, we ask whether or not they have any specific features. And we link to the histone modification. As I mentioned before, histone modification is another important epigenetic marks. And in the past few years, through the chip sequencing analysis, people start to understand that actually histone modification has strong indication of functional elements in the genome. For example, this is K4 trimethylation. This is indication for the promoter region. So basically, I'm telling you here, based on this figure, complete unmethylated CPGs are frequently in the promoter region. And of course, in the complete methylated, this is case 36 trimethylation, that is in gene body or intergenic region. So they have specific histone mark. And now, as you can see from here, cell subset specific methylation, they are highly frequent in the enhancer region, in particular in the active enhancer. Now, if they are in the enhancer, you may think about how about transcription factor binding. So as you can see that we see a bunch of transcription factor enriched in the promoter region or complete unmethylated genomic region, but they are depleted when the genome is completely methylated. But as you can see, interesting, in the cell subset specific methylation regions have some transcription factor. They are enriched, but not all of them. Okay, this work was done for the stem cell. Now I'm going to take something more challenging, is that we are going to try to understand the human brain to see whether we can do anything with it. And then I believe everybody here sitting here will agree with me. Brain cells, they get to have some difference. You cannot imagine that all your brain cells, they are the same, right? That is not, it doesn't make sense to us. But the question is that, what and how they different from each other? Do we know how they different from each other? Is there any way to help us to decode the brain difference, cell difference? So we made an assumption. The brain starts with one cell. Actually, whole body, our human beings start with one cells, right? So if one cell's methylome is like this, then when you look at the multiple cells, you see they are similar, but they are different on some regions, right? They are very similar to each other, but they differ on some regions. So this is the basic, simple concept behind how we're going to decode the brain methylome. So we made an assumption that when you scan the genome carefully, you will find some region become homogeneous, some region are heterogeneous. This heterogeneity in the methylome will reflect the cell difference in your brain. Now we take the brain methylome, and as I mentioned before, the reason we developed a hairpin technique, one of the reasons is to tell whether or not this strand difference, asymmetric DNA methylation or cell subset specific methylation. So we combine these two together. The brain methylome need to say that this is public data set. It's published last year in Science. Now we, as I mentioned before in the mouse stem cell, we found that the cell specific methylation is linked to enhancer. And we also found that the cell-specific methylation linked to transcription factor and affect gene expression. And then I'm going to talk about how that will link to the genetic mutations, human difference. So the first thing we did is to scan the methylome on the human brain and mouse brain. And this have different development stage. So this fetal 35 day, I'll just make sure that you can, if you couldn't see clear. Fetal 35 day, 2 years, 5 years, 12 years, 16 years, and 25 years. So this is development. And this data we took is published in Science last year. So we reanalyzed the data. Based on the Science publication, they found that methylation level is almost staying the same during the development. Methylation overall, whole genome-wide. But we scan them to find a region that show heterogeneity. As I mentioned before, I'm not looking on the average methylation. I'm looking each pattern to see when you're showing a black and white, methylate and unmethylate, and the same in the same regions. And that we calculate the frequency, how frequently we see such variations. We see striking increase in the early development, basically saying that our brain get complicated when we are little. And then it pretty much 2 years old kids, their brain is already quite complicated. And then stay plateau. This is how our brain complexity and the epigenome level tell us. We observe similar thing in the mouth. This is the trend we see. That's basically tell us. Now, the good thing here is that we're able to tell in the genome where these regions have shown at heterogeneity. Now, the next question, of course, are they important? Are these regions associated with any genes? We found that yes. The regions, the genes associated with the cell-specific methylation, they are highly relevant to the brain function. Is neurodevelopment or neurodifferentiation? So now the next question would be, are they enhancers? Yes, we try to overlap the data with the histone modification. Again, here is K27. This is active enhancer mark. But this is data from a Cell paper they published last year from the P0 is the birth, at birth, the mouse. And P7 is 1 week. So we are quite good luck, basically have histone mark and the methylation data at the same time points, P0 and P7. We overlap them together. One is Cell paper data, the other is Science paper data. But interesting finding is that, yeah, they do pretty well. You know, cell-specific methylation indeed enrich for histone modification mark for enhancer. Now, the next question we want to ask, are they have transcription factor binding there? Of course, as you can see, we divide into neuron and glia. They have different patterns. So, and then we are now still working on to dissect the transcription regulatory on this brain development. And the last thing we try to do is that if these regions associated with brain development and so critical, now are they associated with human disease? So nowadays, and then I think today already have quite a lot of talk about the genetic variation. Or in the past couple of days, you hear a lot about genetic variations. So in NIH, they have a collection for all GWAS study, have around 1,000 GWAS study. And this GWAS study collect 1,000 our human disease and trait. So basically, they perform patient and then normal control and to find whether or not have genetic variation associated with specific disease. And when this disease collection, we take this 1,000, we ask a question, this 1,000 kind disease, which kind disease most frequently the SNP will occur in the region we found to be relevant to the brain. As you can see from here, most of the disease find that have a mutation that in the human genome, this mutation is likely to occur on the cell-specific methylation region in the brain. So the addition, Alzheimer's disease, as you can go through the list here, majority of them is relevant to the brain disease and behavior, human behavior. So altogether, I try to give a summary of my talk. I hope that at least my work makes sense to you, is that we're using hairpin bisulfite sequencing technique. We are able to determine the cell subset-specific methylation and are able eventually to distinguish them from the asymmetric DNA methylation. And I also hope I convince you that the mammalian brain development is accompanied with a dramatic increase in the number of cell subset-specific loci. And then the data, we show that cell subset-specific methylation is highly correlated with specific histone mark, and then also enrich for specific set of transcription factors. And the last thing we try to demo is that these regions are critical. It's linked to the genetic variation. So of course, future work will be understand how genetic variation and epigenetic, they crosstalk to each other. And I would like to thank my lab peoples. Dr. Zhao is the one developing the helping technique, and Dr. Mingyang is taking care of all the data analysis, pretty much I show you here. And then graduate students and the postdocs and help me with all these projects. And I also want to thank my collaborators eventually to make this work possible. I'm ready to take your question.
Very interesting talk. Two questions. If you go back to your cartoon, the iPS cells, So this suggests basically, I think the key factor control the differentiation will be the transcriptional level, right?
Yes.
And the epigenetic may be modulate. But the transcriptional factor, different sets make it learn, make it.
I totally agree with that. I need to say that it has been a couple of years debating who is the player, who go first. And then it's now more and more data point too is that the transcription factor initiate, and then they establish the landscape, our epigenome. Therefore, your methylation coming here or the histone mark coming here to maintain the state, to make sure this cell is brain cell and the other cell is liver cell. They are not changing around. So you, yes, maintenance. But you don't want to have environment change, and then you suddenly brain become liver. So that probably is not a good idea, right?
That makes sense.
So I hope I address your question. Yeah. What is your second question?
The second question is I'm interested. You didn't get into the detail of your pattern recognition. So it seems like cell-specific, different cells, has the conserve the region and some of the differentiation. So is that means you have to do the single cell first, the methylation, and then do the pattern recognition? What is your input file?
Yeah. So let's review a little bit about the history. So basically, in year 2000, so we get a human genome sequence done. So we get a human, first time we have the entire human genome sequence. Till now, 2014, we have 1,000 genome project. Okay, finished. I think it's around 3,000 whole genome sequence, public. I don't know, private company, they did a lot of things, don't tell us. But publicly have 3,000. So it means that even that 14 years passed, we only able to sequence thousands genome. I see that in the past couple of years, high-throughput sequencing coming up, next generation sequencing coming up, main driving force, and then we have a lot of sequencing data will come in the next decade or so. But however, I need to say that epigenome is much more complicated. You can imagine that. You can sequence one individual, sequence one out of your cell. I know your DNA, right? But just sequence one out of your cell. I don't know your epigenome because different tissue, they have different tissue methylome pattern. And then need to say that, during the development, during your aging, disease, everything, the methylome is changing much faster than DNA variations that may happen to us, right? So therefore, sequence epigenome is much more challenging. Talking about brains, have 100 billion cells there. Do you want to sequence 100 billion cells? That's just for one individual. So let's put it this way. In our foresee future, it's unlikely single cell epigenome will go that fast. That's why I'm thinking about helping bisulfite sequence technique, why I'm thinking about bioinformatics. We need somebody to do something to move this field forward. Let's say we don't have single cell epigenome now. We do nothing, or we just give up. So basically, the bioinformatics and the helping technique is try to combine together to see whether we can see something now rather than waiting for years later. Yeah. Yes. Yeah. Yeah, so I see your points. You are more interested in the bioinformatics, detail things. We can discuss that later. But one key thing here is that without the bench, I don't trust pure bioinformatics. So bioinformatics basically help you to find the structures. I think maybe go through this slide or make it more clear. So basically bioinformatics can tell you this from them. You can scan the genome, scan the epigenome to identify this region, but you are not sure where they came from. Therefore, I using the hairpin technique to tell that, to distinguish this from that. So now I need to say that we sequence I didn't show you detail here, we sequence the fetal brain, human brain at 22 weeks. And then we also sequence 17 years old brains. And we found that in the human brains, the majority of the heterogeneity we see is cell-specific because the fidelity is 96%. And in the stem cell, we see the methylation fidelity is 88. So basically, my data tell me in the brain, 2 strands, plus strand and minus strand, the methylation pattern are highly similar to each other. So I didn't show you this, probably causing you confusion. So we do need some bench data to prove what's going on. So.
Yeah. How large is your defined region for bipolar region? And did you specifically look at promoter region or is it genome-wide search? Then does it cover genic region as well?
Okay, so the data I'm taking is whole genome data. So basically, I scan the entire genome. So that is first answer. And the second thing is that I need to break down the genome into pieces. You can't sequence the entire chromosome at the same time, right? That doesn't make sense because in a big chunk of region, you may see couple patterns linked together eventually in a chromosome level. So we take 4 CpG size, but I need to say that currently people define differential methylate region using 4 CpG size as a window. So we also using 4 CpG size as a window to scan the entire genome to find this pattern. Does that make sense to you?
So then you link them together if the 4 sites you identified has the same pattern with the next one.
We scan by single CpG. So basically you scan one and then you slide in a window, 1 CpG, and then scan the other one. So it could be in the region overlap. Is that answer your question? Then so 4 CpG size segment, they may overlap.
Then the one with high complexity, DNA methylation complexity, do they usually occur at genic region or potential regulatory region?
In my case here, as I show you, the cell subset-specific methylation is highly enriched in the regulatory elements, in particular in the enhancers. So that is why when the other day we talked about the transcription factor, all this stuff. Yeah. No doubt. I don't have doubt on that. But just the matter is that it's too complicated for us to handle it right now. I mean, it's a challenging question, right? It's a really challenging question to understand the complexity of our brain. So, yeah.